3 researchers who are going to highlight and share their work on delaying cognitive decline. We're going to hear from Dr. Paul Jakes, Dr. Kieran Reed, and Dr. Tong Zhang. With that, let me start introducing Dr. Paul Jakes to everyone. And note that Paul joined HNRCA many years ago and is currently the lead scientist of the nutritional epidemiology team. His research includes B vitamins, whole grains, flavonoids, hydration, diet patterns, and quality. He's published over 300 peer-reviewed manuscripts of original research and has been especially active in areas of B12 and folate related to older adults. He is a well sought after speaker and a favorite professor at the Friedman School of Nutrition Science and Policy. He earned his doctorate degree in epidemiology from the Harvard School of Public Health. And today he's going to open up our webinar with a population level look at Alzheimer's disease and identify some nutrition interventions that may help protect against the development of the disease. So with that, Dr. Jakes.
Dr. Jakes, I think you're on mute.
Okay, thank you for pointing that out. I want to thank you for that kind introduction. This afternoon, what I hope to demonstrate is that the evidence linking diet to Alzheimer's disease risk is compelling, if not yet definitive. But let me first start by disclosing my potential conflicts of interest. This slide lists my research support, which is currently all from USDA or NIH, with the exception of 2 Tufts University pilot grants and a very recent IAFNS lipid committee grant. I am also a member of the DANA North American Essential Dairy and Plant-Based Advisory Board. And with that, I'll start my presentation for this afternoon. And what I plan to do today is to first start by addressing 3 different questions as background. First question is, what is Alzheimer's disease? What is the impact of Alzheimer's disease? That is, why is there so much focus on this disorder? And what are the known risk factors for Alzheimer's disease? Then I'll present evidence linking healthy diets to cognition. Given the limited time I have today, I will not try to summarize all of the evidence relating nutrition and cognitive health. Instead, I will focus on healthy dietary patterns and Alzheimer's disease. I will also discuss some of my own research examining dietary flavonoids and Alzheimer's disease. I'll present the flavonoid findings not only because these natural compounds are key components of healthy diets, but also because they may ultimately help lead to the development of interventions for prevention of Alzheimer's disease, as I will discuss later. Okay. So let me start by describing Alzheimer's disease and its impact. As shown on this slide, dementia is an umbrella term that covers several different conditions that are characterized by a decline in cognitive skills, such as memory, language, and problem solving. Alzheimer's disease is the most common type of dementia, accounting for about 70% of all dementias. There are many changes in the brain of a person with dementia, but Alzheimer's disease is characterized by very specific changes. These include the pathological hallmarks of Alzheimer's disease, the presence of both extracellular plaques composed of a substance called amyloid beta and intracellular neurofibrillary tangles composed of tau proteins. The accumulation of plaques and neurofibrillary tangles can lead to neuronal dysfunction, loss of connections with other neurons, and the death of neurons. Alzheimer's disease is regarded as one of the most important public health challenges in the United States today for several reasons. Tops among them are the enormous impact of Alzheimer's disease on the daily activities and quality of life of affected individuals, the rapidly growing population of older adults, and its significant economic and societal costs. The impact on daily activities and quality of life are due to many factors. We all recognize memory loss as the hallmark of Alzheimer's disease, but there are many other cognitive and behavioral changes that affect daily activities. These include loss of executive function, which makes it more difficult to plan and carry out simple multi-step activities such as dressing, shopping, or cooking, the loss of judgment and attention, which results in poor decision-making, and personality and behavioral changes, including depression and other changes in mood and behavior such as agitation. Finally, Alzheimer's not only affects cognitive function, but also affects physical function. It is a wasting disorder that results in frailty. As shown in this graphic from the Alzheimer's Association, Alzheimer's disease has a very high and growing prevalence and a tremendous impact on mortality. More than 5 million Americans are currently living with Alzheimer's, and that number is projected to nearly triple by the year 2050. Alzheimer's and related dementias are the 6th leading cause of death. 1 in 3 seniors dies with dementia, and the vast majority of those with Alzheimer's. While deaths from many other causes, such as heart disease, have declined over the past 2 decades, deaths from Alzheimer's disease have risen dramatically by almost 150%. Consistent with the remarkable prevalence of this disease, the economic and societal burdens of Alzheimer's disease is staggering. In 2020, Alzheimer's disease and related dementias cost the nation over $300 billion. If no interventions are identified to prevent or slow its development, it is estimated that this cost will grow to $11 trillion by the year 2050. And these costs do not include the efforts of 16 million unpaid caregivers who provide services currently estimated to be worth another $244 billion. Now, Alzheimer's disease is generally classified as early versus late onset or familial versus sporadic. Now, early onset Alzheimer's is arbitrarily defined as a diagnosis of less than 65 years of age. And the early onset disease makes up about 5% of all Alzheimer cases. Essentially, all familial disease is early onset. It accounts for only about 15% of early onset cases, or less than 1% of all Alzheimer's cases. There is little research on nutrition and early onset Alzheimer's disease, and specifically on familial Alzheimer's disease. So my comments today will relate to research based on late onset disease. That said, I don't think it would be unreasonable to assume that some of the findings we see between nutrition and late onset disease, which is essentially all sporadic disease, might also be applicable to early onset sporadic disease. So there's been no lack of effort to identify drugs to treat Alzheimer's disease, but in spite of that, currently there are no effective treatments. Therefore, it is essential that we identify strategies by which Alzheimer's disease can be prevented or its onset delayed. For this, we need to focus on known modifiable risk factors for Alzheimer's, which are listed on this slide. While some risk factors such as age and sex and family history and genetics are not modifiable, they still might provide insight into mechanisms of this disorder. However, our best hope for identifying ways to prevent Alzheimer's is through its known modifiable risk factors. That is, those risk factors that we can change. However, even among the few known modifiable risk factors, we're left with few options for meaningful interventions that might prevent Alzheimer's disease. However, I would argue that the growing body of evidence relating nutrition and Alzheimer's disease suggests that diet should be added to this list of modifiable risk factors. We already know that diet may impact Alzheimer's disease through its effect on cardiovascular disease risk factors such as diabetes, hypertension, elevated cholesterol, and obesity. But evidence suggests that diet also has effects on Alzheimer's disease that are independent of cardiovascular disease. I will spend my remaining time today describing some of this evidence. I will summarize evidence that diet is a key modifiable determinant of Alzheimer's disease by focusing on healthy dietary patterns. So why focus on dietary patterns? Well, dietary patterns overcome many of the limitations of studying the effects of single nutrients of foods. Most importantly, the impact of a single nutrient of food on a health outcome might be small and unmeasurable, but the cumulative and synergistic effects of multiple nutrients and non-nutrient constituents of a diet might result in much larger and measurable impacts. In other words, healthy dietary patterns capture the overall effects of a combination of many dietary factors associated with better health, as well as dietary factors that might be detrimental to health. Given the time limitations today, I will focus on evidence from one dietary pattern, the Mediterranean diet. The Mediterranean diet is based on traditional diets as observed in the Mediterranean basin regions. The Mediterranean diet that is most commonly promoted today is based on the Greek diet from the 1950s and 60s. This pattern is depicted in this graphic on the slide from Always. As you can see, the Mediterranean diet is a plant and fish-based whole food diet with a focus on the use of olive oil right in there. It allows for moderate consumption of poultry, eggs, and dairy, and alcohol, and limits intakes of red and processed meats and sweets. I will now show some of the evidence relating this dietary pattern to Alzheimer's disease risk. This is a slide from the earliest study to examine the relationship between a Mediterranean diet and risk of Alzheimer's disease. It included over 2,000 community-based non-demented individuals from New York. And this slide divides the study participants into 3 equal-sized groups based on the Mediterranean pattern, low, middle, and high adherence to the Mediterranean pattern. These lines represent the proportion of each of these 3 groups that remained free of Alzheimer's disease over the 10-year follow-up. The darkest line represents those who adhered most closely to the Mediterranean diet, and the light gray line, the bottom line, represents those who were least adherent. And at the end of the study participants with the highest adherence to the Mediterranean diet pattern were 40% less likely to develop Alzheimer's disease than those with the lowest adherence to this dietary pattern, a very dramatic effect of diet on Alzheimer's disease risk. This observation was replicated in 6 other studies, but given time limitations today, I'll provide only one additional example. So this slide shows the results of the most recent study of the Mediterranean diet and Alzheimer's disease conducted among approximately 1,000 participants of the Rush Memory and Aging Project. This is a study of volunteers living in retirement communities and senior public housing units in the Chicago area. As in the prior study, participants were divided into thirds based on the degree of adherence to the Mediterranean diet. And by the end of follow-up, the participants in the third of the study sample with the highest adherence to the Mediterranean diet, denoted by the dotted line, were more than 50% less likely to develop Alzheimer's disease than the third adherent to the third who had the lowest adherence to this dietary pattern, as indicated by the dashed line. Again, evidence of a remarkable reduction in risk of Alzheimer's disease. We also have corroborating evidence from a randomized clinical trial, the Prediment trial, that the Mediterranean diet protects against age-related loss of cognitive function. This large clinical trial was designed to examine the effect of consuming a Mediterranean diet enhanced with either olive oil or nuts on a variety of different health outcomes, including changes in cognitive function. The control group was assigned to a low-fat diet in this study. The different colored bars on the slide represent the 5-year change in cognitive function for the 3 arms of the trial, with the lighter bars indicating the Mediterranean diet interventions, and the dark bar, the low-fat control. Bars above 0 indicate improvement, and those below 0 indicate a loss of cognitive function over time. As shown in the slide, participants randomized to the control group, again designated by the darker bars, on average scored significantly worse on cognitive tests for memory, frontal lobe cognition, and global cognition after 5 years on a low-fat diet compared to their cognitive scores at the beginning of the study. On the other hand, those randomized to the Mediterranean diet arms maintained cognitive function or even improved their cognitive scores after 5 years of consuming a Mediterranean diet. As I noted previously, the Mediterranean diet pattern is very high in vegetables, fruits, nuts, seeds, whole grains, and other plant foods. The health benefits of this diet are likely derived from the combination and synergistic effects of many dietary components. However, key components of the plant foods that comprise the base of the Mediterranean diet are compounds called polyphenols. And polyphenols may be key to some of the health benefits of this diet. One large class of polyphenols are the flavonoids. Understanding the role of these bioactive compounds might help in the development of preventive interventions for this disorder. For example, recent evidence suggests that some flavonoids have senolytic activity. Well, those of you who have followed this seminar series from the start will be familiar with this term from the first seminar presented by Dr. Chris Wiley. While my understanding of senolytics is very limited, let me take a short detour here just to present a very simplistic abridged primer on senolytics. So to understand senolytics, I first need to describe cellular senescence, which is a permanent state of cell cycle arrest that occurs as a consequence of cell stress. Senescent cells accumulate in tissues with age, and these cells produce pro-inflammatory molecules which appear to contribute to the development of various age-related disorders, including cognitive decline. Evidence suggests that removing these senescent cells can delay aging and extend health span. That's where the senolytics come in. Senolytics are small molecules that can selectively remove senescent cells. Some natural dietary polyphenols appear to have senolytic activity, including 3 flavonoids, quercetin, fisetin, and luteolin. Evidence for senolytic effects of flavonoids is so far preliminary and based on supraphysiologic doses. However, diets high in plant foods can provide quite large amounts of flavonoids and other polyphenols on a daily basis, over 2 grams per day. While we don't know if daily consumption of physiological levels of these potential natural senolytics will prevent cellular senescence, there is strong evidence that higher flavonoid intakes are associated with lower incidence of many age-related diseases, including Alzheimer's disease, as I'll show you now. I will share some of the findings of our research examining the role of flavonoids in Alzheimer's disease work that was done largely by Ezra Shishtar, a former doctoral student of mine. To examine the relationship between flavonoid intake and Alzheimer's disease, we studied 2,801 Framingham offspring cohort participants with a mean baseline age of 59 years who were free of dementia. We captured flavonoid intake from 5 clinical examinations over a 20-year period. Now, there are hundreds of different types of flavonoids, but they can be grouped into 6 different classes shown on this slide, as well as a class of which is referred to as flavonoid polymers, which are derived from these flavin 3-ol monomers. Given the time today, I will focus on 3 types of flavonoids. First of all, the flavanols, which are derived largely from onions and scallions, apples, as depicted on this slide, as well as kale and broccoli, berries, and tea. The anthocyanins, which are derived largely from berries, the red, blue, and purple berries, red grapes, and red wine. And also the flavonoid polymers, again, which are derived from tea and chocolate, apples, berries, grapes, red grapes, and red wine. There we go. In this slide and in the following slides, we divided study participants into 4 groups based on their flavonoid intakes and compared the risks of developing Alzheimer's disease over 20 years in the highest flavonoid groups, don't denote it here by this symbol, relative to the lowest, the risk in the lowest intake group. And for berry lovers, they'll be glad to see from this slide that individuals in the highest intake of anthocyanins showed a remarkable nearly 75% reduction in incidence of Alzheimer's disease relative to those in the lowest intake group. Individuals in our study with the highest intakes of flavanols, which are derived largely from onions, apples, broccoli, and kale, had a 49% lower risk of Alzheimer's disease relative to those with the lowest flavanol intakes. And finally, for those tea lovers and us chocolate addicts, we saw a significant 47% reduction in risk of Alzheimer's disease for participants with the highest flavonoid polymer intakes relative to those with the lowest intakes. So to summarize and conclude, the evidence may not yet be definitive, but there is certainly compelling evidence that consuming healthy, largely plant and fish-based diets, such as the Mediterranean diet, may help protect against the development of Alzheimer's disease. Plant foods, such as vegetables, fruits, nuts, and seeds, that are the foundation of the Mediterranean diet pattern, are good sources of flavonoids, and evidence would also suggest that flavonoids protect against Alzheimer's disease. And finally, we all know that there are many reasons to consume a healthy diet, including lower risks of cardiovascular disease and some cancers, and now we can add protection of cognitive health and prevention of Alzheimer's disease to that list. Thank you for your attention, and I believe we're holding questions till after the other presentations. Thank you.
Great. Thanks so much, Dr. Jakes. Very much appreciated. With that, let me next introduce Dr. Kieran Reed, who's going to share with us some of his work on nutrition counseling interventions to preserve the independence of older adults. As I'm doing this transfer, there we go. I will note that Dr. Reed is a scientist with the Nutrition, Exercise, Physiology, and Sarcopenia team at HNRCA. He earned his PhD in clinical research from Trinity College, Dublin. And he also earned his master's in public health from Tufts School of Medicine. He has extensive experience and training in conducting clinical trials. And in 2018, he was awarded the Young Investigator Award from the International Osteoporosis Foundation for his community-based engaged study. And with that, as I noted, his talk here today is going to share his work at a community level, which focused on exercise and nutrition counseling for independence of older adults. Dr. Reed.
Thank you, Wendelin. Can you hear me okay and see my screen?
We're good.
Okay, excellent. Thank you for that nice introduction, and also this opportunity to present here today. I'm very excited about this small body of work, and I would like to just say at the outset, it's a little bit different than what Dr. Jakes just presented. This is much more at the sort of individual community level, but nonetheless, hopefully it's a nice segue into some focus on physical activity, specifically for prevention and delay of cognitive decline. I'm just going to make sure I can see my slides. I don't have any disclosures. Okay, I can see my slides now. Okay. So, as I said, I'm going to talk about delaying the onset of Alzheimer's disease and related dementia. But what I'm really going to hope to convey to you is one working model that I am personally and at the center at the HNRCA we're very excited about, is this Engage for Brain Health model. And it's really how we can empower a community to reach older adults with a program of physical activity. And as I'm going to explain to you throughout the course of the next 10 to 15 minutes, how we can really identify and reach an at-risk older adult population. So this is an older adult group that's at risk for developing dementia, but does not have dementia yet. And let me reveal a little bit more about that further on. Dr. Jakes really gave a very elegant overview of the impact of Alzheimer's disease and related dementias on the daily functioning of older adults, their caregivers, the families, and healthcare systems. And we also learned about really the staggering statistics. Dr. Jakes really focused on the US numbers, but worldwide it's pretty similar. In the next 10 years, the prevalence of Alzheimer's disease or related dementia worldwide, it's going to double from 50 million. And by 2050, the prevalence is going to more than triple. And we've seen that these statistics are pretty consistent, especially when we look at the numbers in the US. As we know, there's no drug that can prevent or cure Alzheimer's disease. And just to put this in context, in the last 20 years, at least 150 failed therapeutic agents or therapeutic drug programs have been halted in their attempts to try and prevent or cure Alzheimer's disease. And that's just within the last 20 years. So really, the widespread implementation of effective and scalable low-cost lifestyle interventions to maintain the cognitive independence of older persons should really be an urgent public health priority. And this is being widely recognized now, as you would expect. The World Health Organization released its first guidelines on risk reduction on cognitive decline and dementia, and this is only 2 years ago actually these came out. And as you'd expect, nutrition and in particular physical activity are really high on their list of potential lifestyle interventions that can actually reduce the risk of developing Alzheimer's disease or related dementia. I wanted to build off Dr. Jakes's introduction on the societal and overall costs of Alzheimer's disease and related dementias. And just to show you this really elegant simulation study that was done in 2015, which essentially modeled the theoretical value if there is an intervention that can delay the onset of dementia, the actual benefits that would be both to society and also in terms of prevalence and also to overall cost reduction are really, really emphatic. And you can see here, I'm not going to go through all these bar charts, but you can see the theoretical reduction in a 5-year delay is in this yellow here. So an intervention that is successfully able to prevent or delay dementia for 5 years in 2010, by 2050, this would result in an overall reduction in the prevalence of dementia by about 41%. And approximately, these simulation models have estimated a 40% reduction in the overall total costs associated with this debilitating disease. I really think this is an important aspect to remember, at least from today's presentation, that there is huge, huge value for society if we can actually come up with an intervention that can push this problem down the road a bit. And as I segue into what I think is a potential working model to have some impact on the prevalence and incidence of dementia, I just wanted to share with you one of my favorite pictures of my mother and father, actually. This is looking off the east coast in Ireland, looking out ashore. This was a couple of years after they got married back, I believe, in the 60s. And I just wanted to hopefully share with you today a vision of what we're thinking about as a model. And one of the reasons I want to do this segue from the scientific statistics into some of the working models, components that I'm going to describe is my own family has been afflicted by Alzheimer's disease and dementia. My mother suffered probably for a decade with it. So it really, well aware of the statistics and the overall impact of dementia, but I'm really acutely aware of the personal devastating effects of it on an individual and families and also caregivers. So I like this picture because it shows my mother here with a big picture vision, but I also like the aspect of my dad. He's somewhat focusing in here with his telescope. And this is, I think, what we need to do if we're really going to have some impact here. So let me explain. Here is our working model that I think, and we at the HNRCA could have potential traction in actually delivering an intervention that can delay the onset. We are specifically focused on physical activity, and I am in particular, because there's great epidemiological data supporting the beneficial role of exercise on cognition. There's also some really fantastic data in animal studies, but also some clinical data that shows that when you exercise and have repeated bouts of exercise training, you can increase brain neurogenesis, you can increase perfusion in the brain, functional connectivity, and you can actually maintain or increase the brain volume. And these are all important things for maintaining your brain health as you enter into advanced years. What I also want to describe about the physical activity intervention that we're interested in is how we use an established and a very translatable physical activity intervention that may hold promise for actually being an intervention that can have preventable and delay the onset of disease. We're also focused in this working model that I'm going to describe. We're focused at the community level in centers such as senior centers and places where older adults in the real world spend their time. I'm going to describe our success in actually reaching older adults in a senior center in the greater Boston region. And this is really an important aspect of our model. We think we can identify older adults who do not have dementia yet, but who may be at increased risk for developing Alzheimer's disease and dementia. And the idea here is that if we can identify this phenotype correctly, we can possibly have earlier intervention, and thus the impact of exercise in particular, but frankly, any lifestyle intervention could really have much more impact if we can identify an at-risk phenotype. And finally, one other aspect of the working model that I'm going to show you is we really do want to empower the community agency to effectively deliver the intervention, and in this case, it's physical activity, because this really means we could have widespread scalability in any intervention that we hold true that has some possibility for success. So I'll show you our results of doing that also. And how we've got to this point is actually building off this well-established physical activity intervention that we were part of the life study here at Tufts. And this actual study is the largest study of physical activity ever performed in older adults. It was a 3-year intervention. I don't have time today to go through the major findings, but I included this citation. But this essentially was designed to preserve physical function in older adults. And we actually have previously taken this study and translated it into a senior center in Greater Boston. And it was in the ENGAGE study that Wendelin mentioned at the outset. We were actually targeting mobility and physical performance in this study. But when we went to a senior center, brought the life study intervention, delivered it by our own exercise physiologists at the senior center, we had pretty clear gains in executive cognitive functioning, and this was just after a 6-month period. So this really brought us thinking, well, actual fact, this particular intervention may have potent effects on cognition or preserving cognition, particularly in these more vulnerable populations that are potentially attending senior centers. So we also found that this particular intervention, when we did translate it into the community, was safe and feasible. It did increase mobility, and it also decreased the incidence of falling. So this led us, and just before I go on to the ENGAGE-B study, I just want to describe how simple this intervention is. It consists of walking exercise, which gets the aerobic component of physical activity, increases heart rate and cardiorespiratory function as you're exercising. You can see here the older adults here are performing lower extremity strength training. They're using their body weight to get themselves up off chairs and doing some squat exercises. They're using some ankle weights to perform some lower extremity strength training. And then they do some balance and flexibility training. And this is really the gold standard exercise program now for older adults that was proven in the life study to be effective. And clearly you should understand from this that it doesn't need high-tech, expensive equipment to actually roll this out. All you really essentially need is some chairs and some ankle weights. And what I also want to show to you is that when we think about translating this intervention, we actually think that based on our ENGAGE efforts over the past few years, we can do this activity intervention anywhere. So this is the schematic of the senior center where we did it in Somerville. We actually used the corridors to get these older vulnerable adults walking around and doing 10 to 15 to 20 minutes of aerobic activity. Then they would use the existing chairs and tables to actually do their lower extremity strength training. They did their balance training and flexibility training and some of the ankle weight training here using the existing infrastructure of the senior center. So again, no high-tech equipment needed and no big expense needed to actually have this intervention and host it and deliver it. So this, because of the cognitive findings in our first ENGAGE study, this led us to actually think about, well, how can we target older adults for potential cognitive benefit? And going back to this idea of really reaching older adults who we think can be at increased risk for developing Alzheimer's disease or related dementia. And I'm going to introduce this topic to many of you are probably not aware of Modric cognitive risk syndrome. And essentially, this phenotype was first described by Joe Verghese out of Einstein College of Medicine in New York. And this is essentially an older adult who reports subjectively a memory or cognitive complaint, but who also has slow walking speed. And this combination of these two deficits, it's what makes up the motoric cognitive risk syndrome. And the slow walking speed, it depends on which study we look at, but typically this is a walking speed less than about 0.8 meters per second or so. And as I said, this is really simple to measure and identify, and it avoids the need for complex neuropsychological testing in a clinical setting, for example. So we really can go to the community and identify this motoric cognitive risk phenotype. And I just wanted to show you one of the major findings from Verghese's papers who identified this. The presence of MCR is an early and independent predictor of future cognitive decline and dementia in older adults. And you can see here from this figure, this essentially shows in the blue here on the right, these are healthy older adults. And if you see the green line here, this was the same cohort of individuals who just reported a memory complaint. You see the individuals that had just had slow gait speed. But you look here at the purple line on the left, this is those individuals who had both. They had a self-reported memory complaint and really, really slow gait speed. And this illustrates that these are really at enhanced risk for developing. This is both incident cognitive impairment, but also in the bottom slide is pretty similar, an actual emphatically higher rate of developing dementia over a 12-year follow-up period. And depending on the studies that Verghese and colleagues have done, if you had MCR, you're somewhere between a 3 to 8-fold higher, greater risk of developing subsequent dementia, which is really, really high if you step back and think about it. So we think we have a phenotype that we can potentially identify who are at increased risk of Alzheimer's disease, dementia. But to date, no one has really tried to reach out and actually develop a lifestyle intervention and see is it feasible in this population. And this was really the premise for this new Engage-B pilot study. We wanted to conduct the first feasibility study in this population of a lifestyle intervention and specifically physical activity. So we attempted to recruit 40 older adults and randomize them into the physical activity intervention that I just showed you, or a control intervention of healthy aging education. And this was conducted over a 6-month period. And we did some interim evaluations at week 12 and then final evaluations at week 24. And the objectives of the study is really to figure out, can we recruit community-based older adults with this really at-risk phenotype into a relatively long-term randomized controlled trial? Can they appropriately participate and adhere to interventions? Is it safe? And we quantify that by looking at adverse events rates. Can it be delivered by the existing community-based staff? So this isn't like we did last time, going out with all our well-qualified exercise physiologists and rolling out a program of exercise. This particular Engage-B study wanted to see, could we actually train the existing community-based staff to deliver the physical activity intervention. And the training for this particular study just consisted of a half-day session, and we thought that that's really an appropriate type of training to potentially roll out in a scalable manner in the future. We also wanted to see in this feasibility study the compliance with some of these assessments that we always do in these types of research studies, measures of functional capacity or physical performance. We also wanted to see, could we actually obtain computerized cognitive assessments using an iPad. And we also have some pretty cool measurements of brain blood flow using infrared spectroscopy to actually see, could we get some measurements of what's happening in the brain during physical activity. And this is all occurring in a senior center. So this is some ambitious feasibility aims. They're quite simple if you read them word for word. But what this really is supposed to do is set this up for a much bigger study if we can demonstrate feasibility of these outcomes here. And this just quickly is the study inclusion exclusion criteria. In ENGAGE-B, participants were considered to have moderate cognitive risk syndrome if they met all of the following criteria. Number one, a self-reported memory complaint as assessed on the geriatric depression scale, which is simply, do you feel that you have more problems with your memory than most? We then measure, if they do that, have that memory complaint, we measure their gait speed and have these pre-specified cutoffs, and these are pretty low gait speed scores. They're still able to ambulate and they don't have a dementia diagnosis, and we confirm that with their primary care physician. And with some other criteria here, but all pretty simple. They had to be sedentary, which many older adults are. They also needed written permission from the PCP to participate in the study. And again, this shows you the schematic. We did this intervention in the existing infrastructure of a senior center 2 times per week, about 45 minutes per session. The exercises, as I've shown you earlier, consist of aerobic walking, strength training, stretching, and balance. And importantly, it was delivered by an existing community-based employee. In this case was the outreach coordinator or the health and wellness coordinator at the senior center. Okay, so briefly summarize, despite the early cessation of ENGAGE-B as a result of COVID, our approach and working model suggests that it is feasible to recruit older adults with moderate cognitive risk syndrome for a community-based randomized control trial involving lifestyle interventions, and at least in this case, physical activity. We really can reach a vulnerable high-risk population that are typically not well represented in clinical trials. Older adults with moderate cognitive risk syndrome can safely and successfully adhere to a best practices physical activity intervention delivered by the community agency itself. We can obtain these measurements of cognitive performance on an iPad. We can take measurements of blood flow in this population and the setting. This is really key for our future studies to try and demonstrate larger scale effectiveness. And on that note, we are really thinking this direction now and want to go this route of a larger translational study, particularly of physical activity, but really other lifestyle interventions, nutritional interventions, and other potential modifiers of the ADRD trajectory are really warranted in this moderate cognitive risk population. Finally, I just wanted to leave you with the last word from the participants who complete and participate in these types of programs. And this is just some feedback from individuals. When one individual says when exercising with a group, they're motivated to get there on a regular basis. On their own, they did not really want to exercise. And this just tells us that these group-based activity and interventions that take place at the senior centers or other centers where older adults spend their time are really important, and probably now more than ever. But the idea that you get older adults together in a group is probably the best way to have these effective, sustainable programs implemented. And lastly, despite all the statistics and numbers that I showed you on outcomes, etc., this is a really nice comment from an 80-year-old female participant who, after completing the ENGAGE program, said the pain in her knee is gone and she feels much better and happy, and now she's ready to go and get a boyfriend. So sometimes there's a lot more than just benefits from physical activity on cognition and other outcomes. Sometimes there's some magic that can happen as well in these types of interventions. So with that, I'm going to stop, and hopefully I've presented an exciting working model, or at least a model that we and I are excited about trying to develop further at the HNRCA, and hopefully to bring this to larger populations and other communities across at least greater Boston or New England, and hopefully beyond. Thank you for your time.
Thanks so much, Dr. Reed. Really appreciate that overview. So with that, let's transition to our next speaker today, Dr. Tong Zhang. Dr. Zhang is a scientist with the neuroscience and aging team at HNRCA. Her research studies adult human neural progenitor cells and the potential treatment of brain disease. She's also examining how certain nutrients may provide anti-stress and anti-inflammatory benefits to brain stem cells, and that of course can help prevent brain cell loss during aging. Prior to joining HNRCA, Dr. Zhang was a research assistant professor at the McKnight Brain Institute at the University of Florida. She earned her PhD from the University of Tennessee, and today she's going to focus on how neural stem cells and neurogenesis work, because that creates a baseline from a cellular perspective on how diet and physical activity can improve adult hippocampal neurogenesis, leading to delays in age-related cognitive decline. So with that, Dr. Zhang, please.
Okay, so thank you for the introduction. It's an honor to be part of this wonderful webinar series. Let's see if I can control my slides. Okay. So I don't really have anything to disclose, so I'll go right into my talk. So as Paul and Kieran just talked about, aging comes with a decline in cognitive abilities. And this decline is even more severe with age-related neurodegenerations. So with the declined cognitive functions, the aging individual will lose their ability to function independently and to communicate efficiently with others. Therefore, how to prevent this cognitive decline and improve healthy aging has been one of the top priorities for many of our biomedical researchers. As it was also mentioned earlier, we now know that there's a close relationship between cognitive functions and physical exercise and healthy diet. But the mechanism that's actually lining the effects are still under investigation. So since one of the possible causes for the decline of cognitive functions is related to the reduced neurogenesis in the aging brain, our group has been studying this particular group of cells called neural stem cells in the brain that's responsible for neurogenesis. So in my talk today, I would like to share some of the information on what are the neural stem cells and neurogenesis, and why are they even important to us, and how they're related to cognitive functions. And then we'll look at some of the factors that may be able to regulate the neurogenesis. And finally, share some of our data using blueberry as an example to show that how we may actually be able to increase the neurogenesis with nutrient supplement and hopefully contribute to the delay of cognitive decline and promote healthy aging. So what are neural stem cells and why are we interested in this group of cells? So by definition, stem cells are the cells that can divide and generate all cells in an organ or tissue. And neural stem cell is the stem cells in the nervous system. So it's the fundamental ancestor cells that generate all the cells in the brain. So what neural stem cells can do is to make a copy of themselves, which is the self-renew. This is how they maintain the stem cell pool in the brain. And then they can make a copy of a daughter cell, which is a progenitor cell. And this progenitor cell will differentiate and then make neurons, astrocytes, and oligodendrocytes, which are the three major cell types in the brain. So because of this unique characteristic of neural stem cells, it gives a large, huge clinical application. For example, they can provide resources for cell replacement therapy. They can also serve as delivery tool to deliver therapeutic molecules to the brain to enhance the brain regenerations. So the process of a neural stem cell to generate a daughter cell, which is a progenitor cell, and then to generate neuron is called neurogenesis. This process is very important during the brain development because that's when a small number of stem cells will generate all the cells in the brain and form the whole brain, right? Up until several decades ago, people believed that once the brain is developed, there will be no more new cells generated in the brain, and neurogenesis will stop. However, starting 1960s, scientists start to identify that neurogenesis actually can persist through the adulthood, even into the aging period. However, different from in the embryonic stage, neurogenesis in the adult brain doesn't happen everywhere. As a matter of fact, the adult neurogenesis is defined in two major brain areas. One is the subependymal zone that's lining up the lateral ventricle. The other one is the subgranular zone in the dentate areas of hippocampus. So the new neurons that's generated in the subependymal zone will migrate along migratory stream and goes into olfactory bulb and participate in function related to smelling. And the new neurons generated in hippocampus will mostly remain in hippocampus and participate in cognitive functions such as learning and memory, and participating in mood regulation as well. So because of hippocampus is the one that's closely related to our cognitive functions, the adult hippocampal neurogenesis will be what I'm going to focus my talk on for the rest of my talk. So neurogenesis has been identified in most of the mammals that studied, including rodents, primates, and humans. And it has been now known that it persists through the whole lifespan. However, it is generally accepted that even though a neurogenesis can happen through your whole life, the function of neurogenesis at different point of your life is actually different. For example, during the embryonic early postnatal stage when brain is still developing, the main focus for the function of neurogenesis is to generate new cells and repopulate the brain and central nervous system. But once the brain is developed, the level of neurogenesis will decrease. However, it's still considered very important because it's involved in many neuronal functions such as brain homeostasis, neuroplasticity, tissue regeneration, as well as what we are interested, the cognitive functions such as learning, memory, and pattern separations. It's also important in mood regulation as well. So now it is known that aging has a significant negative impact on the neurogenesis. As a matter of fact, aging is known to be one of the most negative regulators for neurogenesis. The negative impact of aging on neurogenesis include loss of the number of stem cells due to cell death, as well as decreased ability of the neural stem cells to generate new cells. Additionally, it is also known that the pathology of age-related neurodegenerations also contribute to the negative impact of neurogenesis. And then this impaired neurogenesis will in turn contribute to the progression of the disease. Therefore, how to regulate this endogenous neurogenesis has become one of the major targets for developing preventions and treatment for the cognitive aging as well as age-related neurodegenerative diseases. Since neurogenesis is so important, many effort has been put in to study how we can actually regulate this process. As a result, we now know that neurogenesis is a very dynamic process. It's very sensitive to a lot of internal and external stimuli. And some of the factors are listed here, that some of them can increase the neurogenesis, such as physical exercise, as Kieran just mentioned, and healthy diet, as Paul has talked about. And there are factors that actually has negative impact on neurogenesis, which include aging, stress, and neuroinflammations. And among those factors, what we are interested is the nutrition and diet supplement. As summarized in this table, many studies based on animal models have shown that nutrition and diet can play a very important role in regulating neurogenesis, including have effect on the survival of the cells, as well as has effect on the ability of the neural stem cells to generate new cells, which is called proliferation. And among the supplement that people has reported, blueberry is one that our lab has been studying. So the reason we're interested in blueberry is, first of all, it's one of the most common consumed berries by general public. It is very rich in polyphenols, and some of these polyphenols are known to be able to pass the blood-brain barrier and present in different areas of the brain. And they're also neuroprotective because the well-known property of anti-inflammatory and antioxidative stress properties of blueberries. Research from both our lab and other labs have shown that blueberry can actually have beneficial effect on the cognitive functions and neurogenesis in the aged animals. For example, one of our previous study led by Dr. Shuki Hill in the lab has shown that when we feed aged rat with berry supplemented diet, they show an improvement in the Morris water maze test, which is a test for spatial learning and memories. More interestingly, these animals also showed an increase in neurogenesis in the dentate gyrus of hippocampus. And there's a positive correlation between the improvement in the cognitive functions, as well as there's a positive correlation between the cognitive functions as well as the increase in neurogenesis in the blueberry-fed animals. So we're definitely seeing a beneficial effect of blueberries on the cognitive functions and neurogenesis in the aged animals. So what we are interested to see is, can we translate this beneficial effect of blueberries into human cells? So as you all know that there is technical difficulties in how to access neurogenesis in living human brains. As alternative, we propose to study the human neural stem cells instead. Not only because these cells are the cells that's responsible for human neurogenesis, but also because with different types of neural stem cells, we can study the different pathological condition that's related to neurogenesis as well. So in this case, we are studying where we generated the cell from hippocampus of control human beings. So in this case, the cells we actually used is the adult human progenitor cells that derive from hippocampus of control human beings. So these cells are highly expandable. They can maintain in the cell culture for a long period of time without losing the stem cell properties, which make them ideal for studying neurogenesis. So once the cell culture is established, we will treat the cells with blueberry. We'll treat the cell with blueberry for a period of time. And at the end of the treatment, we will stress the cell with, we'll stress half of the cell with cellular stressor to mimic what the stress that the agent can impose on the cells. And then both the stressed cell and non-stressed cell will go through different analysis for different readout. So the first readout we looked at is the viability of the cells, because one of the major negative impact aging put on the neural stem cell is to lose the cell due to cell death. So our data show that when we stress the cell with a cellular stressor, there's a significant decrease in the viability of the cells. However, if we pre-treat the cells with blueberry extract, the viability of both the stressed cell and non-stressed cell will increase, with a significant increase in the cells under stress. We also look at the cell deaths of these cells. So with a cellular stressor, there's a significant increase in the rate of the cell goes through cell death. And if we pre-treat the cells with blueberry extract, the rate of the cell death will significantly decrease, both in the stressed cells and non-stressed cells. Then we look at ability of the stem cell to generate cells, to generate new cells, which is the proliferation. So by labeling the cells with a thymidine analog, we were able to label the cells and quantify them and compare with the control cells. So what we saw was when we stress the cell with the cellular stressor, there's a significant decrease in the proliferation rate of the neural stem cells. And if we treat the cells with blueberry, the proliferation rate of the cells will increase with a significant impact on the cells under stress. So we're seeing a protective effect of blueberries on both the survival and proliferation of the human neural stem cells. So what we are interested is to see what might be the potential cellular mechanism that's underlying this protective effect. So we look at different mechanisms, but with the time limit, what I will focus on today is the inflammation and oxidative stress. Not only because blueberry is well known for its anti-inflammatory and antioxidative stress properties, but also because it is known that with aging, there's an increase in oxidative stress, and there's an increase in the neuroinflammation, and these two works very closely together and significantly inhibit the neurogenesis in the aging brain. And this will in turn contribute to the development of neurodegenerations. So with that in mind, we look at expression of some of the inflammation and oxidative stress markers. As shown here as example are INOS and NOX2. So what our data suggested was when we stress the cells, there's an increase in both the inflammation and oxidative stress markers. But if we treat the cells with blueberries, we didn't see too much of a change. We didn't see a significant change in the inflammation and oxidative stress markers. However, when the cells are under stress, there is a significant decrease in their expression of the inflammation and oxidative stress markers. So as a summary, we think human neural stem or progenitor cells can serve as an important tool for us to investigate the potential beneficial effect of a nutrient supplement on neurogenesis, as well as the mechanism that's underlying this effect. And our data suggests that when the cells are under stress, there's a negative impact on the survival and proliferation of the human neural stem cells. And blueberry can protect this, can protect this cells and revive some of this negative impact imposed by the cellular stressor. So our data suggests that diet can improve the adult hippocampal neurogenesis, and this hopefully will lead to a prevention of cognitive functions and promote of healthy aging. So finally, I want to thank all the members in the neuroscience and aging lab who contribute to this project, and my funding agency as well. Thank you.